Photosynthesis and Cellular Respiration Lab. In accordance with the Virginia Department of Education's Science Safety Standards, students should only watch the following video. Under no circumstance are you to conduct this lab activity at home, regardless what might be said during the video. Before you continue with this lab, make sure you've already read pages 1 through 5 of your lab handout. Pre-Lab Part 1. Separating light using a diffraction grating. Like sunlight, the white light emitted by a light bulb includes several wavelengths of visible light. A diffraction grating separates these different components of visual light. Thousands of closely spaced grooves etched onto the surface of the diffraction grating interact with the different wavelengths of light passing through and diffract the waves at different angles. Each separated wavelength of light corresponds to a different color. Here is an example of what a diffraction grating would look like. Use this example to answer question 4 in your lab handout. Pre-Lab Part 2. Relating colored filter results to photosynthesis. Photosynthesis, the process by which green plants convert light energy into chemical energy, is dependent upon the input of solar radiation. Plants initially absorb light within the visible light spectrum, which ranges from 400 to 700 nanometers, in order to begin the light-dependent reactions of photosynthesis. Plants and algae utilize several different pigments, such as chlorophyll A, chlorophyll B, and carotenoids, that absorb different wavelengths of visible light. Examine the absorption spectrum measurements of these three chloroplast pigments in Figure 3 on page 2. The pigment absorption measurements were collected using an instrument called a spectrophotometer. The absorption of light at specific wavelengths by plant pigments drives the light-dependent reactions of photosynthesis. Use the information provided in Figure 3 to answer questions 6 through 11 in your lab handout. Lab Part 1. Observing indicator color change. In this beaker, I have a mixture of water and phenol red. Now, phenol red is an indicator, meaning it indicates certain levels of pH. Now, this phenol red is red at a pH of 8.4 or higher and turns to an orange or yellow color at a pH of approximately 6.8 or lower. What I'm going to do is I'm going to breathe into this straw, and that straw is sitting inside the solution. And I want you to carefully observe what happens to the color of this solution. After you've observed what happens, there are 4 or 5 questions in your lab handout that you need to answer that reference what happened through this observation process that you're about to undergo. Here we go. Notice how our solution has changed from a red to an orange. Now that you've seen this color change, answer the questions in your lab handout that follow this example. Lab Part 2. Linking photosynthesis and cellular respiration. Use this diagram to help you answer questions 5 and 6 in your lab handout. Question 7 says, fill in the table below based on your current observations of the indicator experiment from Lab Part 1. Use the indicator guide below. The indicator that we'll be using for our lab experiment today is bicarbonate indicator solution. As you can see, the 2 most important bits of information from these charts that you need to gather and pay attention to are the stock solution pH values and the bicarbonate indicator color. The chart at the top of your screen gives you the word form, and the chart at the bottom of your screen gives you an actual representation of the color of the indicator solution at each of those pH values. Lab Part 3. Using algae beads as a model organism. For this experiment, we'll be using algae beads to test for the processes of photosynthesis and cellular respiration. As you can see, algae beads are small green spheres. Each sphere is filled with thousands of algae cells. For this experiment, I filled 3 microcentrifuge tubes with approximately 0.7 milliliters of water. In 2 of those tubes, I placed around 6 algae beads. In all 3 of those tubes, I placed approximately 5 drops of the bicarbonate indicator solution. So now I have my experiment set up. I have my control that just has the bicarbonate indicator solution. It doesn't have any algae inside of the container. I have my container that has no light being exposed to it. So I've got the container that has the bicarbonate indicator solution along with some algae that's completely covered so light can't penetrate to the algae beads. And then I have my third example or my third experimental setup where I have the bicarbonate indicator solution along with the algae. Now, if you look on page 14 of your lab handout, you're going to see a big chart, and it has the time, 0, 5, 10, 15, 20, 25, and 30 minutes. And then you have the bead with light, bead without light, and tube without beads. At the end of each of these time periods, you're going to need to document the color of the tube as well as the approximate pH of the tube based on the chart that was shown to you earlier in this lab. Time, 0 minutes. Based on the color of the bicarbonate indicator solution, determine the pH for all 3 trials. Here are the pH value colored standards to help you make your observation. Time, 5 minutes. Based on the color of the bicarbonate indicator solution, determine the pH for all 3 trials. Here are the pH value colored standards to help you make your observation. Time, 10 minutes. Based on the color of the bicarbonate indicator solution, determine the pH for all 3 trials. Here are the pH value colored standards to help you make your observation. Time, 15 minutes. Based on the color of the bicarbonate indicator solution, determine the pH for all 3 trials. Here are the pH value colored standards to help you make your observation. Time, 20 minutes. Based on the coloration of the bicarbonate indicator solution, determine the pH for all 3 trials. Here are the pH value colored standards to help you make your observation. Time, 25 minutes. Based on the color of the bicarbonate indicator solution, determine the pH of all 3 trials. Here are the pH value colored standards to help you make your observation. Time, 30 minutes. Based on the coloration of the bicarbonate solution, determine the pH for all 3 trials. Unfortunately, this picture was unable to properly document the color of the bicarbonate indicator solution in the light experimental setup. The color of the bicarbonate indicator solution in this setup for the light experiment was actually a dark purple. It was not this dark brown color that's shown in the picture. Here are the pH value colored standards to help you make your observation. Lab Part 4, Post-Lab Questions. At this point in the lab, you should have collected all of your data. Now you're going to need to analyze that data and answer the accompanying questions that follow in Lab Part 4's Post-Lab Questions section of your lab handout. Thank you for watching this video.